So hi, before I go into my presentation, I just wanted to thank everyone on behalf of myself and the rest of the GSS committee for there being so many people in attendance this morning. It's a really good opportunity for some of us that haven't had the chance to really speak and practice public speaking again, because I haven't been in this situation since undergrad, so thank you for being here. So I'm going to talk about some of the methods my lab and I are using to explore the data seen at these little black dots and use those to explain the eddy's impact on biogeochemical structure in the Southern Ocean. A little motivation behind the research: why the Southern Ocean? Not only is it just a really cool region in general, but it's really important factor when we start thinking about things like climate regulation, global ocean nutrient transport, the uptake of CO2, and a number of more things that I haven't listed up here on this screen. And so one of the things we can study is biogeochemical properties in the Southern Ocean as indicators of a changing climate. However, it's an incredibly difficult and dangerous region, so it leaves us with these really large data gaps in these biogeochemical properties. And with those large data gaps, we have a lot of unknowns when we're trying to understand how the Southern Ocean can be impacted by a changing climate and therefore the things that it factors into. So many of these gaps are filled oftentimes with very large scale models that use really, really coarse data. And an example of one of those, what one of those models looks like is up here. The next picture will be a higher definition model because it includes eddies in that resolving, in the resolving background of the model. And this is just one example of many studies that are recently coming out that are reflecting how when you add eddies into these Southern Ocean models, we're still not getting the perfect picture to be able to fill in those data gaps, but we've never been more accurate. So we're starting to get there. And so by adding eddies, these mesoscale vortices that are smaller than the grids that these models use, these become more accurate because these eddies have been found to have a variety of influences on biogeochemical processes. So using sea surface height anomalies, you can spot either cyclonic eddies you're going to see in the blue, and those are typically coinciding with cold cores and upwelling. Anticyclonic eddies, on the other hand, you're going to see a positive sea surface anomaly, and those are usually associated with warm cores and downwelling. And I say usually because these are extremely variable physical properties. In addition to these temperature variations and the vertical motion, eddies can influence nutrient transport in a number of ways, including eddy-induced Ekman transport. You can have, in addition, air sea fluxes that are motivated by the spin of the eddy, and then you're going to have different fluxes depending on that. Again, a more variable topic. And then you can also have things like eddy trapping, where those biogeochemical properties are physically trapped within the eddy and can trap as the eddy moves. Or you can have eddy stirring, where those properties are then mixed with the properties around it. So a lot of variability in terms of biogeochemical properties. And what does that mean to everybody here? Well, a statement from McGillicuddy 2016 suggests that the impact of these eddies on biogeochemical properties can impact everything from the lowest trophic level to the top predators, especially in this region. Not only are they super important, they're super abundant. So a study from Frenger et al. 2015 found that there were over 1 million eddies identified in the Southern Ocean between 1997 and 2010, suggesting that there is an even greater impact because of the sheer number of these eddies that are available. So one of the ways we're starting to fill in those data gaps are by using autonomous vehicles. For example, what we're using with our data from BGC Argo floats. And these have been able to reach regions and seasons that we haven't been able to sample in before. These floats collect data by drifting at about 1,000 meters for 9 days. They drop to 2,000 meters on the 10th day and then rise up to the surface. And as they rise up to the surface, they collect temperature, salinity, pressure, and biogeochemical properties at discrete depths, send it off to the satellite when it reaches the top, and restarts that cycle where it drifts, goes down, collects a profile, sends it off. And we take those profiles to understand in really high resolution what's going on both inside and outside of eddies, because these floats have been observed to be inside immediately in eddies, on the outskirts of eddies, really far from eddies, everywhere in between. So we can use that to try to understand how these eddies might be impacting the biogeochemical properties in different regions, seasons. That's the motivation of the project. So the null hypothesis we're trying to refute is that there are no differences between the impact of anticyclonic eddies, cyclonic eddies, and non-eddies across all seasons, basins, and frontal zones on biogeochemistry in the Southern Ocean, trying to refute this. And we're going to do that by looking for what I'm going to define as eddy differences. And so In really basic terms, what I'm going to call different is when profiles from one type of eddy lie outside of the standard deviations of another. And so this plot is not our actual results. It's just an example of what we'd anticipate to see. But it reflects that an anticyclonic eddy and a cyclonic eddy have standard deviations, which are the dashed lines that are outside of range of each other. So they are providing different impacts on the biogeochemical property, in this case, oxygen. And we can use that to compare not only different types of eddies, different seasons, different frontal groups, and I'm just going to go over what that organization of that data looks like. We take satellite altimetry and profiles from our BGC Argo floats and separate all of our data into anticyclonic, cyclonic, and non-eddy profiles. We then break those subsets into our seasons, winter, spring, summer, fall. We then break those subsets into the basin, so Pacific, Atlantic, and Indian, and then we break down further by frontal zone. And I'm going to show you what those frontal zones are in a minute. I highlighted in red here water masses because we haven't yet, or not yet, reached that point in our analysis. And you're going to see that it's really important that we do, and we do it soon. So what it looks like when we break all that data down is, for example, these are cyclonic eddies in the Indian Basin in all seasons, so all cyclonic eddies. I'm going to break it down. I've chosen winter for this example, and then I've broken down by frontal group. And what I mean by frontal group, for the purpose of this presentation, knowing exactly which fronts I'm talking about is not important, but knowing that the space in between the fronts, which are these lines in the Southern Ocean, each have their own unique biogeochemical properties. So I can't average over the entire Antarctic circumpolar current. I have to go between the fronts of it. So frontal group 1 is going to have different properties from frontal group 2, which has different properties from frontal group 3. So you have to break it down in multiple different ways to be able to actually see where the differences lie and isolate it to whether or not it's from cyclonic or anticyclonic eddies. And so we find those differences by calculating anomalies. And we're currently using a baseline where we calculate a mean from, we take a variable. For this example, it's going to be oxygen from non-eddies. And we calculate a mean at each depth through every single profile that matches a non-eddy within a certain frontal group, within a certain basin, within a certain season, a lot of repetition. And we calculate a mean for this example for spring, non-eddies, in front group 1. I've left basin out of it just to avoid repeating myself over and over and over. And what you'll notice is that there's a lot of spread and variability just within this one plot that is likely skewing our mean already. And that's going to come in handy when I start talking about some of our first results. We take that mean from non-eddies as our baseline, and we want to remove that signal from our cyclonic and anticyclonic profiles. In this example, I'm just going to work with cyclonic. We take an individual cyclonic profile that matches the same frontal group, season, and basin, subtract that mean non-eddy profile, and we come up with an anomaly profile. Those anomalies are what are plotted here. And then we take a mean of all of those anomalies and the standard deviation of those anomalies. Those standard deviation and means are what we use to compare and look for where differences between eddies would exist. And so what we're getting as our results, those don't look very different. So there's two really important things to point out here is that when you look at cyclonic in blue and anticyclonic in red, the means themselves, the solid lines in the middle, they look relatively different, but their standard deviations do not. They overlap each other almost exactly. The second thing to look at is those standard deviations. We expect there to be a lot of variability because eddies can impact biogeochemistry in a number of ways, but this spread is too big. And so while our conclusions say we're not currently able to refute that null hypothesis, we're aware of some limitations that exist. And one of those major limitations is that bullet point I had highlighted in red, which was water masses. And so we believe, if I go back to where we calculated our mean and our baseline that we're trying to remove signal from, if you look, you can see kind of a pattern of some outside variability that doesn't fit, specifically between 0 and 500 meters. And then again, from 500 meters down to 1500, you see a whole nother signal. And we believe that those are different water masses that are also within that frontal group. And those water masses are going to have, yet again, different biogeochemical properties that are then going to impact the mean that we're trying to use. And these signals are then replicated also through our cyclonic and anticyclonic eddies. So they are also, those profiles are also observing different water masses as they go through different frontal groups. Going to be to use biogeochemical data from pre-existing climatologies to locate where within those frontal groups these water masses are. And then we're going to recalculate anomalies based on creating a subset by those water masses. And hopefully we'll be able to see a little bit more difference between the spreads available in the difference of impact from cyclonic to anticyclonic over seasons, basins, frontal groups, and water masses. I wanted to thank my really awesome team, Dr. Chambers, Dr. Tamzit, Dr. Williams, and Sarah Barloni, there she is, for their really awesome support on both our lab project and mine, because they overlap quite a bit. And then thank you to the National Science Foundation, fellowships from USF, and the SOCOM project for a combination of funding and really sick data. Either or.
Nicola, great talk. So what does it mean? So like, let's say if you find that the cyclonic and anticyclonic eddies
Sure.
don't significantly differ, like,
Right.
what do we what's the bigger conclusion from that?
Sure. So it kind of goes into, I guess, the purposes of why this portion of research is taking place. And I have two different reasons. So for my personal research, I am looking at one float in particular that has a set of daily profiles. And I'm going to use whether or not we find significant differences to determine whether or not we see significant daily variations and compare it to what these, because these profiles are 10-day temporally spaced. So I want to try to use this data to support what I may see when I look at one-day variations. And it could very well be we don't see it. And hopefully the way that we're doing it is being able to isolate it and say eddies alone are a reason. And if we can't say that eddies are, based on the variability we see, we can say it's likely something else, and that needs to be looked at. For our overall lab project, this is serving as a kind of base foundation for looking at CO2 fluxes. That kind of goes into some of the background of where air sea fluxes can be influenced by eddies in the area. And so the next step would be look at what do CO2 fluxes look like and what we would deem as different eddies. And that's sort of to go through an overall calculation of maybe a Southern Ocean carbon budget and how to think about what we're adding into the atmosphere in terms of, or what we're adding to the atmosphere is glossed over with Southern Ocean models. And so how can we kind of motivate that you need to improve those models? And that's where that CO2 flux goes in. And this is the background to kind of support why we need to do that. Yeah.
So your multicolored oxygen profiles all seem to be very nice curvatures to them. But at 1,500 and 1,600, I think it was meters or pressure, you have these really sharp shifts right there.
Yeah.
And I don't understand where those come from compared to the, oh, maybe there is a shift in those data. Yeah.
There's a shift, and that's a great question, and that is something that we are currently looking at as well. If I could say this is very preliminary data,
Sure.
That would probably be the best explanation. So there's a multitude of things outside of just our biogeochemical plots that we're looking at to try to make a more smooth application of this. But one of the things that we're hoping is when we go to the climatologies and narrow down to water masses, that from that stage, we might be able to remove some of this noise. We think it could be noise, but it is an ongoing process and still kind of formulating our methodologies to be able to come out with an anomaly that we feel really good about.
The variability, have you considered for your basic parameter instead of a mean, have you considered medians? Because, you know, it's the old, if Bill Gates walked in here, you know, we'd all be millionaires by
right.
On average, but
I don't have the support for that, but that's a really good idea, and that's something that we could actually very quickly run through and see what we're seeing. In terms of means, we were going off of some very small previous studies that kind of stopped before getting to that final answer. And that was sort of the approach, was to use means. When we bring in the existing climatology data, we're going to test and see, is that an actual thing we should be using as our baseline? So medians will be something to look at when we bring that in as well. Thank you.
All right.
Oh my God. Unless I can take it later.
So in the 80 is a different sizes.
Yes.
Also 80, the water property, temperature 80 profiles within different part of the 80. For example, center of 80 and on the edge of 80,
Mhm.
very different.
Yes.
So when you did sort out this cold 80 and warm 80 or cyclic and anti-cyclic 80s, did you use a certain search radius?
Yeah.
For 80s?
Yeah. So
Or
we.
you need data first?
Yeah, so when we are trying to locate eddies, we use surface altimetry, and then we have kind of a toolbox that finds those anomalies, and then we set a radius of kind of like a boundary of what we're willing to call inside of our eddy. In terms of exact location, where some floats might be dead center and some might be towards the outskirts, and there's going to be, again, differences in the properties there. We haven't dealt with specific locations of profile within an eddy. We just have, yes, it's in an eddy, or no, it's not. And I know that's something that wants to be done, but is going to be outside of the scope of these projects, because these projects are all going to serve as motivations to do more science having to do with eddies in the Southern Ocean. Yeah. Thank you.
All right.
Thank you, Nicola. Our next speaker today is one of the newest students here at CMS. Angelique Rosa Marin is joining Pam Halleck-Muller's lab, and today she's going to be talking about forams as bioindicators of reef health.
Good morning, everyone. Thank you, Catalina, for that wonderful introduction. So today I'm going to be presenting some of my master results from Florida Agricultural and Mechanical University. So to start, I just want to acknowledge like all the people and also entities that were involved in this massive project. Sea Grant, CCME, they funded me to basically do my master, my old lab, and also a bunch of people that helped me in the field, in the writing, and through the whole career. So with that in mind, my project was based in the Hobos National Estuarine Research Reserve. That's in Salinas, Puerto Rico. And this reserve, it's part of the 29 National Estuarine Research Reserve Systems, alias the NER. And it's the only reserve located in the Caribbean, okay? So with that in mind, as I mentioned, the reserve is located in the southwest part of Puerto Rico. You can see it there, that wonderful picture. That's the whole estuary. And right now, the bay is confronting some threats, mainly globally, right? Climate change, ocean acidification, and also sea level rise. And with that in mind, they are looking for cost-effective and also rapid monitoring tools that they can apply to their management plan in order to better understand their coral reefs, health, mangroves, and also the bay, inside of the bay itself. So how my project, of course, like try to add to this situation is that basically I was proposing to use benthic foraminifera assemblages as a valid indicator of water quality changes and also environmental parameters, with the idea of use them as a bio monitoring tool for coral reef ecosystems. So basically the whole hypothesis of the project is to environmental parameters will show seasonal changes, and this variation hopefully will be reflected in the foraminifera assemblages. So my study site, as I mentioned before, Hobos Bay, Puerto Rico. I work specifically in these reefs right here. And in this tip of this reef right here, their names are coming soon, boom. Cayo Morillo, Cayo Morillo, Cayo Pájaros, and Cayo Caribe. Basically, as you can see in the pictures, we delineated back reef and fore reef transects. We had in average between 2 and 3 stations in transit. And at each station, we collect and measure environmental parameters. We collect water, superficial water samples, and also sediment samples. This was a 2-year-long project, so we have data from 2018 and 2019 as well. And to have data that represent the wet and also dry seasons, we collect samples in March, August, September, and also December as well. So going back to the lab, I measure nutrients, orthophosphate, ammonium, using mainly colorimetrically methods, sorry. And then with the sediment parameters that I measure with the superficial sediment samples, I analyze for grain size, organic carbon, and also calcium carbonate. And of course, the portion to count my farms. So moving now with the results, we can see here in this box plots temperature here in the right and salinity in the left. We see very clearly here the expected seasonal fluctuations in temperature and salinity. And we also saw higher salinity in March, which is one of the driest months in Hobos Bay. So, So for pH and dissolved oxygen, we saw lower pH concentrations in warmer months, which was August and September. But basically very consistent pH in the dry months, which were March and December. And in terms of the dissolved oxygen, this graph here, we see fairly consistent oxygenation through all the samples. Now with the nutrients, orthophosphate and ammonium, in overall for both nutrients, we have very low concentrations, which is great. I mean, it was coral reef, so it was the expected concentrations for oligotrophic environment. However, we have these peaks here in Cayo Caribe and Cayo Pájaros. And these reefs, well, both reefs are super close to local communities. So this can be by anthropogenic influences and the human activities around the estuary. And with the nitrates and also nitrate, similar situation, we saw very, well, higher concentrations of nitrates in comparison with phosphate and ammonium. But we also have these big concentrations here in both, and it could be mainly through terrestrial runoff. That's also one of the big things right now that the reserve is experiencing. It's been entering a lot of undesired chemical components and also pollution through land-based and terrestrial influences. And now here we have organic carbon and carbonate calcium. I just want to make a note here in this part here in Cayo Morillo. For the carbonate calcium, we found very high percentage of the carbonate calcium. And this is a very shallow and small reef that it's super exposed to wave energy and also wind influences. So me in the field, from my observations, I found a lot of coral fragments, and so probably that's why we have that biggest concentration in the carbonate calcium. So moving now with the sediment texture, also the grain size. This is a summary of the sediment texture of each reef. Here inside of the bay, I also call it like the lagoonal area. It was mostly dominated by finer grains. It was a lot of muddy and stinky sediments. And then in the fore reef here outside, Cayo Morillo and Cayo Pájaro were super consistent with these bigger grains, coarser and also gravel grains. However, here in Cayo Caribe, we also found a lot of finer grains as well. And now moving on with the foraminifera assemblages, the exciting part. Here we have the four-reef for marsh, the four-reef top dominant taxa that we found. And here we have the back reef. So in the four-reef, we see this species here, Amphistellaina, being the predominant. And in the back reef, we see Discorvis and Ammonia and Ephirium being the dominant species and what that means. So basically, Amphistellaina is very well known to be a good water quality organism, and it's found a lot of reefal environments. And species such as Discorvis, Ammonia, and Ephiriums, they are small forms. So they are heterotrophic, and also they are very well known to be associated with stress environments, right? So in August and September, in the four-reef, we still see Amphistellaina being the protagonist of the movie. And then in the back reef, we still see these species that, again, are heterotrophic groups. And then in December, similar situation, Amphistellaina being dominant in the four-reef, and in the back reef, we still have Amphistellaina here, but we also have those heterotrophic species that are basically indication, and they love to be in finer grains and potentially like fluctuations in the water quality. And in the foraminifera density, in this graph here, we have the density in the y-axis and the reefs in the x-axis. And now, well, in this graph, as you can see, the fluctuations of the density per reef and also per month. Basically, we notice a lower foraminifera density in the summer months and higher variability in Cayo Morillo. And this also could be because of the wave energy and wind influence that potentially is sorting more the sediments. Now here in this graph, we saw the same situation, but divided by the four reef stations and the back reef. And basically, in the four reef stations, we found fairly a lower density of forms in comparison with the back stations. And because of this density situation, it was a little bit challenging to make interpretations of the area, because in some stations I found like 6 or 10 farms. So those stations were a little bit, it was complicated to make an accurate interpretation about that area. So with that in mind, I'm closing this talk. Basically, we saw how the water quality and also environmental parameters reflect the seasonal changes in Hobos Bay, and also it reflects good water quality conditions in the reef area. And we also saw how the benthic foraminifera assemblages reflected that, especially having Amphistellaina, which is again one of the good water quality indicator species, being predominant, especially in these four reef locations. And as a conclusion, I mentioned a little bit water depth and water energy, especially in Cayo Morillos and Cayo Pájaros, which are the shallower and more exposed reef. It seems to be the two main controlling factors for the foraminifera assemblages in that area. And for further interpretation, and actually that's a great catch, and that's where some of my PhD work, it's gonna be looking at like how the foraminifera density and also reef depth limit, right, that interpretation. So basically my PhD now, I'm gonna try to look how we can sort that out, basically. So thank you so much. If you have any questions, I'm happy to answer.
No questions, I'll ask a question.
Right.
I was just curious about the wave energy concept you're bringing up, because on the southern side of Puerto Rico, you're far away on the wrong side of the island in terms of receiving large ocean swells. And so do you think it's more of a catastrophic type of event related to tropical storms that would cause this change, or do you think it's just the periodic wind waves?
Yes and no. Yes, because my data, I have data before and also after some tropical storms, and also after Maria, which is one of the biggest hurricanes that Puerto Rico went through. So probably, yes, it can be do these natural phenomena. But I mean, from me being there in the field, that area of the southwest part of Puerto Rico is extremely windy. Like in a good day, it can be 15 to 20 knots. And most of my field work, it was that was a limitation. So so yeah, that and actually that is why I'm interested now in looking more on that aspect of the wave energy and also currents. That location in the south part of the Puerto Rico, it has a lot of currents influence. So that's something that I plan to work on more. Yeah, whichever, I don't know.
So you said Vista was an indicator of good water quality.
Yes.
I was wondering what you mean by that. Like
Yeah,
what
course.
kind of parameters are you talking about?
Yeah, of course. I mean, a little bit of background of forums, and also Amphistegia. Amphistegia, it's what we call it a symbiont bearing forum. So basically, it has a very similar behavior to corals when they are exposed to similar stressors, such as like fluctuations in temperature, fluctuations in salinity. They tend to behave similar to corals again, so they tend to digest or expel their symbionts. And they also can even show bleaching. So it's very well known through the literature how this form is well associated with rainfall environments. So that answer a little bit your question? Yeah. So basically the rationale is that if we have that farm there and it's not bleached, it's in good condition, we can make the assumption that the conditions of that reef are suitable for reef survivorship or potentially further development.
Thank you.
Yeah. There was a, yeah.
Looking landward from your site, it seems to be very agricultural.
Yes, oh yes.
And that seems to mesh with your results that during the rainy season you see very high nutrients.
Yes.
Does the preserve have a good relationship with agriculture, or in that whatever results you find, would they be willing to work to reduce?
Oh yes, of course. Yes. And adding on that thought, Hobos Bay, I mean, that area, it's being even like historically impacted. We had a sugar mill in the 80s, I think. We have an active power plant there. And yeah, in the 80s and 90s, those lands were super well used for agricultural purposes, and they still do in some areas. But yeah, answering your question, I think, or comment, yes, the reserve is super very well known about these issues, and in my interactions with them, yeah, it seems to match my results with the current anthropogenic activities in the area.
All right, I think we'll have to move on right now, but
Yeah.
you can talk
Thank
after,
you.
sorry. Thank you, Andy. So our next presenter today is Callan Crawford. Callie is a master's student in Kristen Buck's lab, and today she'll be presenting on labile nickel in the North Pacific Ocean.
You can also like actually click a link.
Okay, all right, so this. Yeah, not sick.
I've got You can go. All right. Thanks, everyone. Thanks for the introduction, Catalina. Again, my name is Callan Crawford, and today I'm going to be presenting my master's thesis proposal titled Labile Nickel Concentrations Across Biogeochemical Gradients
in the North Pacific Ocean, insights into nickel bioavailability. So to take a step out, I want to talk about the importance of trace metals overall. We know that trace metals are elements that are present in very small concentrations in the oceans, usually on a nanomolar or 10 to the negative 9th scale. However, we also know that they're important for macronutrient acquisition. And so what I want to point out is on this figure we have pulled from Morel et al. 2020. We see a biomass or like a phytoplankton cell in the middle, that's the green mass, and then it's surrounded by 3 different macronutrient cycles on the outside: carbon, nitrogen, and phosphorus. What we see in the middle are these trace metals and their use in various different enzymes. Those are the gray boxes. And we know more about some of the more heavily studied trace metals, such as iron and copper, but what we know less about is some of the understudied trace metals, such as nickel. So the importance of nickel is that it's used in 2 major enzymes, the first of which is called a superoxide dismutase, or SOD, and this importance is highlighted in the green box here. SODs are used to hydrolyze reactive oxygen species, or ROSs, that are produced during the electron transport chain into their non-toxic counterparts, and so they're used for cell protection. The other enzyme that nickel is used in is called urease, and this is used to uptake a certain type of dissolved organic nitrogen called urea. The general form for urea is highlighted in this red box here, and we can see that nickel's use in the urease enzyme allows phytoplankton cells to uptake and grow on urea. So when we talk about nickel profiles and when we see nickel profiles in the ocean, this is a very classic figure from Brulin and Franks, 1983. We have depth in meters on the y-axis from 0 to 4,000 and nickel concentrations in nanomolar from 0 to 12 on the x. And I'd like to point out 3 different trends here. The first trend is that we see nickel having a nutrient type profile, which means that it has lower surface concentrations compared to those in the deep. The second trend is that the deep Pacific concentrations are greater than those in the deep Atlantic due to the age of the water masses. And both of these trends support a classic nutrient type profile. So this is, when we think about a nutrient type profile, all of the nutrient type elements will have these kind of trends. The thing that distinguishes nickel from other trace elements is this persistent 2 nanomolar in the surface ocean. And we are not entirely sure why this sticks around. So we think that it might be tied to its bioavailability, but we're not entirely sure. So we can't talk about bioavailability without talking about chemical speciation. And this just refers to the different chemical forms that an element will take in seawater and can be described using this equation here. So we can see on the left-hand side, we have nickel concentrations, or the total dissolved nickel concentrations in seawater. And this is equivalent to 2 different terms. The first term is this nickel prime term, which is the sum of all the inorganic species. These are all considered to be labile, and this includes the most bioavailable form, nickel 2+. Nickel 2+ is the most bioavailable form because it can be directly taken up by the cells by binding to ligands that are embedded into the cell membrane surfaces. And then the second term is this NIL term, and this is referring to the organically complex nickel. This is generally assumed to not be labile and not bioavailable, but we think that a portion of this might be, which might explain this 2 nanomolar in the surface oceans. But this is much more difficult to calculate when we compare it to the inorganic species, because the inorganic species, we can take hydrographic data, kind of throw some math together, and we get a number at the other, on the other side. The labile nickel is much harder to measure directly. So because of this difficulty in measuring labile nickel directly, there have been very few, when I say very few, I literally mean these 2 that I'm about to show you, labile nickel studies in the open ocean to date. So the first one is from Saito et al. 2004. We can see that this group took 2 different transects in the Peru upwelling region shown on the map to the right, and they produced this data in the figure on the left. In this figure, we have nickel in nanomolar from 0 to 10 on the y-axis and longitude moving from east to west on the x. And we have total dissolved nickel in the black circles and dissolved labile nickel concentrations in the open white triangles. And what we can see is that for almost all of the stations that have dissolved labile nickel concentrations associated with the total dissolved nickel concentrations, there's about a 1 nanomolar gap, if you squint a little bit, that is persistent kind of throughout this transect. But we think that, we can see that most of this total dissolved nickel is actually labile. Now, when we look at the second study, which is from Boiteau et al. 2016, we see a similar transect also off of the west coast of South America, but that they extended this transect well into the Pacific Ocean, into the oligotrophic gyre region. And their data is in this bottom figure here with, again, dissolved nickel on the y-axis from 0 to 4 nanomolar and longitude from east to west on the x-axis. And they see a very similar trend where we have this distinction of about 1 nanomolar between the total dissolved nickel and the labile nickel. So this is interesting that we're seeing very similar trends from two studies that were relatively far apart, but from the same study area. Now, because of these lack of other studies and lack of field data, my first aim is to collect the first depth profiles of labile nickel in the open oceans, and then examine how the portion of labile nickel changes compared to total dissolved nickel with differing depth and biogeochemical regimes. So my first hypothesis is that concentrations of labile nickel and total dissolved nickel will have nutrient type profiles. As a reminder, we're looking for low surface concentrations and higher concentrations in the depth. And then my hypothesis 2 is that concentrations of labile nickel to total dissolved nickel will reflect biogeochemical regimes, and we suspect that the system that will have the lowest labile nickel will be the oligotrophic gyre system. So this is a map of my study site. I was on the RV Sikuliak for a month out in the Pacific last June. It was a huge blast, but you can see the transects that we took. We started from station 1, and they're numbered numerically, and these cover all 3 of my distinctive biogeochemical regimes. So station 1 is representative of a coastal upwelling system, station 5 is representative of an oligotrophic gyre system, and station 8 is a high nutrient, low chlorophyll region, and is also Ocean Station Papa, which is a very well studied site. So to measure labile nickel, I'm using a method called competitive ligand exchange, adsorptive cathodic stripping voltammetry. Try saying that 3 times fast. And it's split into 2 different sections. The first is the CLE section, which is where I add a competing or added ligand. In my case, I'm adding dimethylgloxime, which is DMG, to compete with the ligands that are already present in a sample. So we can see that this creates a new mass balance, which is the first equation that I'm showing you. And you can see this competition in kind of this set of equations below, where this L prime is representative of the ligands that are naturally occurring in the sample, and the DMG prime is the GMG that I've added that is not complex yet to the nickel. Once everything equilibrates, we have this new nickel DMG complex that forms, and this is what we measure or use in the second part, the adsorptive cathodic stripping voltammetry piece. So the new nickel DMG complexes that have been formed are then adsorbed onto a hanging mercury drop electrode. And then we run a potential through the cell, which quote unquote strips this hanging mercury drop electrode of the new nickel DMG complexes, and this produces a measurable current. So we can see the data that we get directly off of the instrument here. This is called a voltammogram, and we can see current in nanoamps on the y-axis and potential in volts on the x. And this is a series of voltammograms laid on top of one another. And what I'd like to point out is that we can see a change in peak height that is corresponding to the concentration of nickel. So to measure total dissolved nickel, which as a reminder is the labile portion plus the non-labile portion, I'm using 2 different instruments. The first is pictured on the left, and it's called a CFAST system, and the system is used to pre-concentrate samples to make them easier to run on the second instrument, which is pictured on the right, which is our element XR mass spectrometer here. This is some of my preliminary data. Again, this is a depth profile, so we have depth in meters on the y-axis and labile nickel concentrations in nanomolar on the x. And this is my data from station 5, which is the oligotrophic gyre system. So my hypothesis 1 was that labile nickel would have a nutrient type profile. This doesn't look exactly like the nutrient type profiles that we think of classically necessarily, but because it's preliminary data, I think it's okay for now. And we can still see kind of these lower surface concentrations in comparison to the greater concentrations at depth. So this is very exciting. So to conclude, just a couple of reminders that nickel is important in 2 different enzymes, urease and the superoxide dismutase. It has a nutrient type profile in the ocean, but in comparison to other trace element type trace metals, we don't see this drawdown to 0 in the surface ocean, and we are not entirely sure why, but we think that collecting these first set of profiles in the ocean across these different biogeochemical regimes will fill a really large and important knowledge gap that we have. So, oh, sorry. And these are all the lovely people and organizations that I need to acknowledge. I'm sure I'm missing some, but I can now take any questions.
No one's going to ask a question. I noticed you had a lower drop down in nickel on your preliminary data than you saw from the South Pacific. So I'm just curious.
Yeah.
You're down to almost 1 there. So that's an interesting finding, isn't it?
Yeah, it definitely is. It definitely is. And, you know, classically, we think of nickel having this 2 nanomolar in the surface oceans. I know that there probably is a study, I think I'm probably forgetting which study it is, but it might go down to like 1.8 or something like that. So I don't think that it's totally unheard of, but generally we find that it sticks to the 2 nanomolar-ish. But yeah, it's interesting to see kind of that data point. Voltammetric measurements are notoriously very finicky, and I was also doing this on a moving ship. So, so you know, this is preliminary data, as I said before, and we're definitely looking into some of these variations. I've analyzed depth profiles from some of the other stations, and they are not as pretty as this one. So there's definitely some variation that likely occurred, and we're going to try and kind of sort that out. So it's really interesting to think about maybe that this is like a real kind of drawdown that we're seeing, or maybe something that was just instrumental. So to be determined. Yeah, absolutely.
So you said that you're using the XR and the CFAS, and you're using this, what is it, cathodic?
Stripping, well, yeah. Yeah.
So this method here, the CFAS and the XR,
Mhm.
you're using that to measure the total nickel?
Yes, absolutely.
And then you're parsing out the labile by using
The voltammetric measurements. Yep, that's absolutely correct. So the voltammetric measurements are run on either a Metrum or a BASI instrument, and these are, well, when we go back, you can see me using the BASI system here at sea. So if we look at, like, this is the hanging mercury drop part of this. There's actual real mercury in this. That was kind of scary. And then it's kind of blocked by the ropes that we have that were like jerry rigged to the ceiling. I don't know. It was a trip. But this is the cell that I'm running all of my instrumentation and measurements in, and that's where the solution and all the chemistry occurs.
Is that the deal with the ship roll?
Sorry?
Is it hanging from the ceiling to a deal with the ship rolling?
Yeah,
Okay,
absolutely.
yeah,
Absolutely. Yeah. So, you know, if the instrument like shakes or vibrates or tips over, obviously that's not good. But yeah, we're trying to minimize the amount of like excess noise that we see in these voltammograms, because, you know, this is a presentation. I'm showing you the pretty ones, but there are ones that look way worse than this. So yeah. Okay, thanks.
And it is my pleasure to announce the last speaker of this morning. It's Catalina Riano. She's a master student working with Aligram, and she will present today on her field adventures at sea. And while she was at sea, I was taking care of the kitties.
Well, hello, and thanks for hanging around for the last talk. I'm going to try and keep it as light as I can. It's not as heavy on the science as the other one. So I had a really cool opportunity to go on a pair of research cruise experiences last year that, by complete coincidence, took me to almost exact opposite sides of the Atlantic Ocean near the poles. And I'm a little sad I'm talking about the poles because I'm already really cold right now, but it's okay. So I titled my presentation Postcards from the Poles. So my first cruise took place in May of 2022, and it took me from Tampa to Newport, Rhode Island up here. And that's where I met up with the NOAA ship Okeanos Explorer. We left out of Newport and sailed for about 5 days towards the Charlie Gibbs Fracture Zone, which lies at about 52 degrees north, 34 degrees west. This cruise was part of a NOAA internship called the Explorer and Training Program. And it involved a 4-week stint on the Okeanos with work focusing on seafloor mapping and hydrographic survey. The cruise I was on, EX2204, was the first of 3 cruises that made up the 2022 voyage to the Ridge Expedition. The goal of this expedition series was to collect baseline information about unexplored and poorly understood deepwater areas of the Mid-Atlantic Ridge. So to achieve the goal of answering some of these questions, well, the scientific questions that guided this expedition included: 1, is the uneven terrain of the Mid-Atlantic Ridge suitable habitat for deep sea corals and sponges? 2, what role does the Mid-Atlantic Ridge play in transatlantic biological connectivity? And 3, what communities exist in the water column above these habitats? And to play its part in answering these questions, my cruise, 2204, had the sole purpose of being a mapping or reconnaissance cruise. So the main data product that we gathered was multi-beam bathymetry, but we were also collecting acoustic data of the sub-bottom geomorphology, as well as the water column habitats of the Charlie Gibbs Fracture Zone. So a little on the study site, the Charlie Gibbs Fracture Zone is a section of the Mid-Atlantic Ridge, which you can see here. It's where a pair of transform faults has extended into approximately 2,000 kilometer long scars in the neighboring crust. Like all mid-ocean ridges, it's the site of seafloor spreading with volcanism and hydrothermal venting. It's also an area where subtropical and subpolar water masses converge, creating a high nutrient region that sustains a high biomass of zooplankton. And for this reason, it was established as a marine protected area in 2010. So the types of data collection that were performed to support and meet these goals included the continuous acquisition of multi-beam bathymetry, water column data, sub-bottom profiles, and sound speed profiles. And I'll come back to why we collect sound speed information in just a second if you're not familiar with it. So all the work I did happened in this room here. This is the Center of Scientific Operations on the Okeanos. It's the mission control room. It's really cool. And I sat at this desk right here the whole time. And the majority of what I did basically was processing multi-beam data. Less formally known as killing dots, because what we're doing is looking for noisy data points in this point cloud and deleting them. Along with cleaning data, I also helped make daily products, which were essentially cleaned and completed maps of the coverage that we got that day for bathymetry, backscatter, and sub-bottom. And getting back to what I mentioned before, I collected sound velocity profiles, which are a fundamental part of mapping with multi-beam sonars. So sonars work, most of you probably know, by sending out acoustic waves through the water column that reflect off the seafloor and then are sent back to the sonar receiver. Because the propagation of sound through water is highly dependent on the temperature, salinity, and density of the water, all of which can vary a lot in the ocean, it's really important to be aware of how water masses are changing and affecting the data that we collect. So without accurate sound velocity, your bathymetry won't look good. You'll get these like arcs in your data, basically. They call them smiles and frowns, rather than an accurate representation of what the seafloor should look like. And there's lots of ways to measure sound velocity profiles, but what we did on this cruise was use XBTs, which are pictured right over here. And these are expendable bathythermographs. And we used an auto launcher like this one pictured up here, which you could launch from the control room. Unfortunately, XBTs, I don't love the fact that they're disposable. You throw them overboard and they stay at the bottom of the ocean. But the way they work is that they fall through the water column collecting temperature data, and from that you can derive the speed of sound through the water column. So the final result of our cruise was that we collected 46,250 square kilometers of new high-resolution data, bathymetry of the Charlie Gibbs Fracture Zone. And this image here that NOAA released shows where we mapped, and it highlights really well the difference between satellite-derived bathymetry on top, and in comparison to high-resolution multi-beam bathymetry that we gathered. It also illustrates really well why we need initiatives like Seabed 2030 to map the world ocean to high resolution, because while satellite bathymetry can give you those really nice big picture maps that you see, you're not going to get this kind of resolution, which is needed for things like navigation safety, habitat mapping purposes, among other things. We also found, which this was pretty cool, we found what appeared to be a lot of bubbling in the water column. So this is the water column image right here. And it correlated with sites along the spreading center that had these kind of like circular dome-like features. And we speculated that this could have been the site of hydrothermal venting. Unfortunately, they weren't able to go back on the next cruise to dive on it with ROVs like planned because of bad weather. However, they did get to do some dives further south, which produced some really cool and interesting follow-up. So EX2205 sailed south of where we were. They deployed the ROV, which confirmed the presence of these deep sea corals that exist along the steep terrain of the Mid-Atlantic Ridge, which is one of the questions they had posed at the beginning of the expedition. This image here on the top right shows the bathymetry. The way they do it, they basically map the night before and will plan the ROV track. It's really hard to see, but the ROV track goes up along here, which is pretty cool. On the same cruise, they found this really cool volcanic pillow mound near the Azores Plateau. And it was found to provide habitat for a variety of organisms, including corals and sponges. And finally, on the third cruise, EX2206, they dove on a fracture zone similar to Charlie Gibbs, but further south near the latitude of Miami. I thought it was an interesting analog. There are different oceanographic conditions there, so it's not an exact comparison. But they found some really beautiful animal life down there, including hydrocorals, sponges, sea stars. So that was pretty cool. And although my cruise was not an ROV cruise, we did ask very politely if we could see the ROVs, and they were nice enough to uncover them for us. So the main ROV is the one here on the left. This is the Deep Discoverer, and it's what produces NOAA's really famous, like, beautiful deep sea footage. And this one right here is its companion ROV, Sirius, and it basically hovers above Deep Discoverer to give you really cool bird's eye view shots of it. The ROVs are piloted by and belong to the Global Foundation for Ocean Exploration, with GFOE. So they have their own independent team on board just for ROV ops. This is Chris right here. He's from GFOE, and he was super cool and just showed us everything, and he was super excited to show us. And that brought us to the end of the cruise. And a quick shout out for anyone who may have met her or who hasn't met her yet. Maggie is one of our newest students here, and she was my roommate on this cruise, so I hope everyone welcomes her as she settles in. So fast forward a few months from my cruise in the North Atlantic. In mid-November, my second cruise took me from Tampa all the way to the southern tip of Chile, where I met up with the German research vessel Polarstern. And from here, we sailed due east towards the subantarctic island of South Georgia, which lies at about 54 degrees south, 36 degrees west, which is nearly a mirror image of where I was in the summer, which I thought was really crazy. So I took part in this cruise as a continuation of work that Ali and his colleagues began to study the glacial history of South Georgia, as well as the modern day role in which the island's biogeochemistry plays in climate change. And there were quite a few goals to PS1332. Some of these included understanding the sources and transport pathways of iron and other nutrients, trace elements, and carbon compounds into the waters of South Georgia and on into the Antarctic circumpolar current, and particularly the role that methane seeps play in the introduction of these compounds. We also wanted to refine South Georgia's glacial history since the last glacial maximum. And we wanted to fill in data gaps with high-resolution bathymetry and subsurface sediment profiles for a better understanding of the gas system around South Georgia. And of course, there was a lot more that I couldn't fit here. In total, there were 13 scientific groups, so it was a really busy, really interdisciplinary cruise. The basis for our work were previous cruises to the island, which discovered hundreds of methane seeps. These seeps were discovered in the troughs and the fjords that were carved out by glaciers. It's believed that the source for much of the methane is basically its organic runoff from the island, which was buried, decomposed, and then re-released as a potent greenhouse gas. So understanding and quantifying the sources of methane in the ocean, particularly the Southern Ocean, where less is known about this process, is really important to a more complete understanding of climate change. So to achieve the goals of this cruise, the hydroacoustics team that I was a part of used an array of sensors. We have first and foremost the HydroSuite multi-beam sonar, which is a really cool specialized sonar designed for use in icy waters since the polar stern is an icebreaker. There was the Parasound sub-bottom profiler, which allows you to look at the upper layers of sediment on the seafloor, and the EK80 echo sounder down here, which is used to survey the water column. So something really interesting happened on this cruise. One of the main goals was to locate some of the hundreds of methane seeps that they had found on previous cruises. But for the entirety of our cruise, we didn't see a single one of them on our systems. We did see plenty of evidence of it, however. So this picture here from the Parasound, you can see this kind of like jagged edge on the bottom of all the sediment layers. This is called gas blanking, and that's what you get when gas is accumulated in the sediment. So we were seeing that, but we weren't seeing the actual bubbling of the flares. And initially, everyone was just like really confused about it, and they were basically theorizing that maybe it was a seasonal phenomenon. But just last week, I actually found out they were there all along. We just didn't have the correct settings in place to see them while we were underway. So yeah, right here, these are all the bubbles that we were going over every day, apparently. So it's a really good lesson if you're a hydrographer. Despite missing out on the gas flares, we still got plenty of good work done. On the left here is our cruise track. And on the right, in color, these are all the areas that we mapped, including 2 hydrographic surveys that we did in troughs on the north side of the island. We also did several Parasound surveys that were conducted independently of the hydrographic surveys to identify suitable sites for gravity coring. And those are the sites that are marked by the stars. So once, let's see if I can get this to play. It's a really cool video. It's very choppy. But so once sites were identified for gravity coring, it was prepared with the desired length of iron piping and the inner plastic liner, and it was deployed over the side of the ship. So this is a shot from the really beautiful Cumberland Bay. So once the cores came up, what we did, we cut the cores, we capped them before the sediment came out the end, which it especially wanted to do if there was gas expanding in it. We would run them inside, label them, and then store them in a cold container. And this was definitely the more demanding work of the cruise, much harder than just sitting behind the computers for hydrographic survey. And you core at whatever time of day is suitable, and sometimes this was in the middle of the night, and we were running back and forth carrying heavy cores. So it was hard, but it was really fun. Later in the cruise, we would open the cores by splitting them in half, and you would keep one half for archiving and the other half for sampling. Sampling was really fun. I enjoyed it because we got to wear cute little blue astronaut suits. And interestingly, because of the location of South Georgia and the time constraint that we had, we only were there for about a week and a half. So most of the sampling that we did happened after we left South Georgia, on our way to Cape Town, where we finally went into port. Some other work that took place included sampling with the multi-core here on the left, which takes clusters of shorter cores at the sediment-water interface. The sediment geochemists were doing pore water sampling. The biologists were doing some really cool bottom trawling. They pulled up these amazing giant brittle stars and some other really cool stuff. There was even onshore sampling. They went onshore to get sediment, water, and organic material. And this brought on some really curious onlookers. Physical oceanographers, they took handheld CTD casts right up to the front of the glacier to study the plume of glacial runoff. And they also did standard CTD casts, which is what we derived our sound velocity profiles from. So we got a really special opportunity on this cruise to set foot on South Georgia, which doesn't happen very often. So it was really exciting for all of us. They took us to shore at the old Grit Beacon whaling station, which was abandoned in the 1960s. You could still see the structures, the ships, even whale bones scattered everywhere here. It was really cool, or crazy. And we also got to see the local residents up close, like this baby fur seal, the adult fur seals, and the very plentiful elephant seals. They were mostly females all over the place. And then we actually came across the beach master, which is like the technical term for the male elephant seal that controls all the females in the area. Thankfully, he was asleep the whole time. It was very startling when we saw him. But yeah, even the seals were all over the place. They didn't really move out the way when you walk by. You can see right here, it's just content to lie there. The female elephant seals were just making these really funny snorting noises the whole time. It was great. It was really awesome. And there were also the king penguins, which are kind of like mini emperor penguins. They're really adorable. And back here, these buildings are, this is King Edward Point, which is the research station for the British Antarctic Survey. It's like one of, or if not the only, like place where humans live on South Georgia, I think. Of course, we completed the visit with the pilgrimage that every scientist must do when visiting South Georgia. We took a hike to the gravesite of Sir Ernest Shackleton. And from up there, we got a really nice view. This little dot right over here is the polar string. So overall, it was really just an incredible experience to part of the world that I may never get the chance to visit again. So I'm super grateful to the incredible people I got the chance to sail with. And I'm especially grateful for Ali, who got me on this cruise. Even though he didn't make it on there with me, he was supposed to, I made sure to carry on the lab's name very proudly, because at this point, it's becoming something of a tradition. This is, I think, around 2010, Ali on the Polar Stern. This is me just now, and Rosemary, our lab mate, is currently on it until like March. So with that, I want to say thank you, and if you have any questions. So yeah, it was more for the experience of the NOAA stuff. Well, yeah, none of it really relates to what I'm doing here, but the stuff for the Polar Stern cruise is relates to what Ali studies. So yeah.
Just to follow up on that question, how about around St. George's Island? I mean, you were at water depths that would have related to paleo shorelines, correct?
300 meters. Yeah, 300 to 150. Yeah. So I mean,
maybe
technically, but yeah, those out there, we mostly were seeing like glacial moraines and yeah. All right. Well, thank you guys so much for coming to the oral presentation. Come back for the presentation.
Thank you, everyone. We're going to take a break now for lunch. Lunch is served in the MSL lounge, so feel free to grab whatever, and then we'll see you all again at 1:30 for the closing presentations. We're going to have another drawing at 1:15. As long as your name is in here, you win the prize. So it's not your calling because you're visiting and you're not that.
Jeff.
Yeah. We can do.
Thank you.
Yeah. Oh, shit.
Yeah.